can be broken down into three domains: bacteria, archaea, and eukarya. Seeing that the physical structures were not enough to classify organisms, biologist Carl Weiss et al. Looked at the intricate differences in ribosomal RNA and have found vast differences between those microorganisms. Thus, the traditional five kingdoms of life, bacteria, protists, fungi, plants, and animals, were put into three domains: bacteria, archaea, which look like bacteria but differ genetically, and then finally placing the protists, fungi, plants, and animals into domain eukarya. Bacteria and archaea are made of prokaryotic cells, lacking a nucleus and membrane-bound organelles, and to date, no true multicellularity exists in these groups. Eukaryotic cells contain both nuclei and membrane-bound organelles, and many eukaryotic organisms are multicellular. What's interesting about Domains Bacteria and Archaea is that they've been around for 3.9 billion years and had a planet to themselves up until about 1 billion years ago. So if there is life out there in the universe, we can certainly bet that they are microscopic. Why? Not only are they very simplistic, but we can find them everywhere. And I mean everywhere. Think of the lamest love song used to measure how much you love somebody. They're found on the tallest mountains. You can find bacteria in the deepest trenches of the ocean, volcanoes, ice, and extremely toxic hot springs. Look, if your love for someone needs to span the extremities of Earth, your relationship is unhealthy. If someone truly loves you, they don't want you to prove it at the expense of your own life. Finally, microbes are present in and on you. I don't care how much you love to be alone, you are never alone. There are at least 10 times as many prokaryotic cells living on and in you than cells that you are made of. But listen, don't worry. Most of them simply love to take up residence because you are warm and moist. Ugh. You know what? Moist is one of those most hated words, so let's just bleep that out of my videos from now on. See, it worked. Anyway, only a few microbes, like less than 1%, are dangerous to human health. So let's get started with the history, diversity, anatomy, and importance of prokaryotic microbes. Before the 1980s, all prokaryotes were lumped together as bacteria. But with the introduction of advanced genetic techniques, we have found that some of those bacteria are actually a separate group called archaea. So where do these things come from?
Earth is about 4.6 billion years old. Wait, how does one know how old the Earth is? Well, this is based on the age of meteorites, since they are not subject to Earth's geological forces. You see, many of the rocks we see today are younger than Earth because they are subject to erosion, heat, and pressure. Meteorites originate in space and do not have these pressures and are much more preserved. Every meteorite ever tested turns out to be around 4.6 billion years old, and since the entire solar system, including Earth, was made at the same time, Earth is 4.6 billion years old. For the first 500 million years, no life existed because the fundamental components used to sustain life were just not available. That is, until 3.9 billion years ago. In one special location, pools of water with the right temperature, still very hot by today's standards, and contained nutrients. This allows small molecules like water, carbon dioxide, and nitrogen to form into larger molecules like carbs, lipids, proteins, and amino acids. Over time, with more heat and sunlight, these form the first cells. The first life forms were made from these cells and did not have oxygen present. These are termed anaerobic organisms. Some species of these still exist today, and due to the harsh conditions of Earth at that time, microbial mats formed which are a lot like coral reefs, but made of microbes. This provided protection to underlying prokaryotes from the harsh elements of Earth at that time. These microbes are held together by a gelatinous substance. Because photosynthesis did not exist yet, many of these microbial mats likely got their energy and nutrients from nearby hydrothermal vents. These vents are found deep in the ocean where a fissure occurs, releasing chemical energy sources. And yes, there are microbial mats still found near deep-sea vents today. See the pattern? Science makes these explanations much on what we see today. In some parts of the world, fossil stromatolites can still be found. And these are made of ancient microbial mats and are aged to around 3.5 to 3.9 billion years ago, which is our best estimate of the age of life on Earth. Early life lived under extreme heat, pressure, and pH. All of these conditions would kill most eukaryotic life forms. Thus, biologists labeled these microbes extremophiles. And to this day,
Extremophiles?
can be found in volcanic areas, hot springs, ice, and really salty environments. Extremophiles also give credence that life exists outside of planet Earth. Strange thing, however, is that these extremophiles could not survive in the comfortable settings that we like. Eventually, some of these anaerobic organisms could undergo photosynthesis, converting sunlight into food. A special group of bacteria called the cyanobacteria evolved the ability to produce oxygen as a waste gas, thus increasing oxygen in the atmosphere, which would be important for multicellular life forms later. Prokaryotes have simple anatomy consisting of plasma membranes, cytoplasm, DNA and RNA, and ribosomes for the production of proteins. Many have cell walls which provide protection as well as shape to the organism. While prokaryotes come in many shapes, the three most common are spherical, rod, and spiral shape. Prokaryotic cells do not have membrane-bound organelles and thus have no nucleus. They have a single circular chromosome twisted into a nucleoid region. All cells have membranes, and most prokaryotic cells have a cell wall with varying composition, which I will discuss later. Some prokaryotes have an additional layer outside the cell wall called the capsule, which allows the bacterium to attach to surfaces and prevents dehydration. And some prokaryotes also have a flagellum or a pilus for locomotion and attachment to surfaces. If you've ever had a stomach ulcer, you have felt the flagella of Helicobacter pylori boring into the lining of your stomach. In addition to a chromosome, some prokaryotes have a plasmid, which is extra genetic material. The best studied plasmids are those related to antibiotic resistance. Prokaryotes encompass both domains Eukarya and Archaea, but what differentiates them? Physically, the composition of the cell walls and the lipid membranes. Bacteria and eukaryotes have phospholipid membranes, while archaeans have a different composition altogether. Also, when comparing DNA between both groups, they differ vastly, indicating that they split from each other early on in the history of life. And just like in plants and fungi, the cell wall in prokaryotes protects the organism and gives the cell shape. However, the chemical composition of this cell wall is made of peptidoglycan. The cell wall in bacteria is so important in medicine as a way to identify specific species of bacteria. Generally, bacteria can be divided into two groups, gram-positive and gram-negative. This is done by using a staining technique that colors gram-positive bacteria purple and gram-negative bacteria red to pink. This coloration is a reaction to the stains based on the composition of the cell walls in each group. Gram-positive bacteria have a thick peptidoglycan cell wall made of many layers outside of their plasma membrane. Examples of gram-positive bacteria include species that cause strep throat and staph infections. Gram-negative bacteria have a thin layer of peptidoglycan sandwiched in between two layers of membranes. Escherichia coli, or E. coli, is an example of a gram-negative bacterium. Archaeans also have cell walls, but are made of a different molecule called pseudopeptidoglycan. Bacteria do not undergo mitosis due to having no nucleus. The nucleoid, shown in red, copies itself and distributes itself to opposite sides of the bacterium. It then develops a septum to divide the cells, allowing the cells to break off of one another. Some single-celled eukaryotic organisms also undergo binary fission as a means to asexually reproduce. While this and cellular mitosis can lead to making more cells, all the cells are unfortunately identical to each other. However, the key to life is genetic diversity. More diversity of genes amongst the population increases the chances of a species to survive changing environmental conditions. But how do living things achieve that, especially prokaryotes? Well, since sexual reproduction doesn't exist in prokaryotes, meiosis can't happen. So how does this happen in this group? Well, the three most common ways in which prokaryotes acquire new genes is transformation, which happens when a prokaryote takes up free DNA in its surroundings. Conditions have to be ideal for this to occur as genetic material is quite fragile. Transduction can occur when a virus, which has a protective protein coat, acquires DNA from another species and then injects it into the DNA of a prokaryote. The third one is called conjugation, and this happens when one prokaryote forms a hollow tube called a pilus, and this pilus is then used to transfer genetic material to another species.
Perhaps when most people think of bacteria, they think of the devastating diseases caused by them. Examples include tuberculosis, typhoid fever, Lyme disease, syphilis, strep throat, MRSA, various infections, leprosy, tetanus, botulism, stomach ulcers, cholera, and gonorrhea. There are many more, but surprisingly only around 1% of existing bacteria cause disease in humans, and no archaeans to date cause disease in other life forms. Of course, back then, we blamed disease on evil spirits, but through science, we understood that in fact microbes can cause disease. In fact, one important field of medicine is germ theory, which explains disease transmission and isolating the cause of a disease down to a specific species.
Disease records go back as far as 3000 BC, with pandemics causing significant declines in populations of people. That's right, pandemics are actually a normal part of human civilization. Boy, did humanity get lucky these past 100 years between Spanish flu and COVID-19. By the way, those last two are viruses, not bacteria. Bacterial pandemics include typhoid fever, which wiped out 25% of Athens, Greece's population in 430 BC. The Justinian plague caused by the bacterium Yersinia pestis near the end of the Roman Empire, which may have wiped out almost 50% of Europe. And the most devastating pandemic to date was the Black Death, another plague caused by Yersinia pestis. It decreased the estimated world's population from 450 million to 350 million people. That is 25% of all of humanity. It was spread by the bacterium residing in fleas and being carried by rats. And since people were disgusting back then, rats multiplied, causing the disease to spread. Europeans developed resistance to many bacterial and viral diseases, but unfortunately carried them over to North America, where they wiped out sometimes around 90% of Native American populations. Believe it or not, North America had a huge Native American population and was not as isolated as you might think. And since Native Americans were isolated from the Old World for thousands of years, they did not develop the immunity against these diseases, and much of that culture and history is now lost. With better sanitation, isolation, and medical treatments, such as antibiotics, death from infectious disease has dropped dramatically. But disease still kills more people in the 20th century than all wars fought in that same time frame. I'm sure many of you at one point has had a bout of food poisoning. These are caused by food-borne pathogens that make you feel like death for several hours with continual vomiting and diarrhea. Botulism is a food-borne disease caused by the bacterium Clostridium botulinum, which results from improper preservation of foods such as packaging and canning. In canned foods, an anaerobic or oxygen-free environment is made, which is ideal for this species. Sterilizing cans and jars before use and cooking at well above boiling temperatures, as well as adding things like citric acid and other acidic substances, greatly reduces cases of botulism. Salmonella can be contracted by contaminated poultry, eggs, and peanut butter. But most cases of foodborne illnesses are now caused by animal waste coming into contact with food. While E. coli is mostly harmless to people, with some strains living naturally in your body, E. coli 0157H7 has led to some fatal illnesses. This is usually linked to eating raw or undercooked meat and eating raw vegetables exposed to animal and even human feces. Most foodborne illnesses can be prevented by cooking meat to proper temperatures and washing your produce thoroughly. As mentioned previously, most prokaryotes are not actually harmful to your health. In fact, without many prokaryotic organisms, eukaryotic life could not function. They assist in digestion, help provide nutrients, and help in decomposition. Prokaryotes are also integral to our economy as a means of making many of the foods and beverages that we love. Another benefit to prokaryotes is bioremediation, which is the use of prokaryotes to remove pollutants. Examples of bioremediation has been used to remove agricultural pesticides and fertilizers from groundwater, toxic metals like selenium and arsenic, as well as reducing mercury from its ionic form, Hg2+, to a neutral and non-toxic form, Hg0. Mercury is an ingredient in many pesticides as well as the production of batteries. Unfortunately, toxic mercury makes its way into the environment and accumulates into the tissues of organisms. As you move up the food chain, mercury accumulates, which is why predatory fish like shark and swordfish have higher levels of mercury than lower level fish like tuna and mackerel, which is why it's not recommended to eat high amounts of predatory fish. Bioremediation has been exceedingly useful in cleaning up oil spills. Mostly, specific nutrients are added to an area of an oil spill to promote the growth of bacteria that can degrade hydrocarbons, which petroleum has lots of. There are even bacteria that have been genetically modified to degrade oil itself. These methods are so efficient that an oil spill can be degrading by 80% in 1 year.
We are not singular organisms, but walking ecosystems housing hundreds of prokaryotic species. Some of the bacteria that reside in our bodies have a commensalous relationship with us. This means that the bacteria benefit by being in a nutrient-rich, warm environment, but we are neither benefited nor harmed by their presence. In fact, you probably don't even notice these microbes in your body until something goes wrong. There are some bacteria that are imperative to our health. You may have heard of the importance of gut bacteria. That's because gut bacteria have the ability to break down certain foods that we cannot on our own. You can also blame some of those bacteria on gases that are released from the digestion of food. They also can help absorb certain ions in the colon and help make certain vitamins. In addition, bacteria help build and maintain the immune system by acting as training targets to our own white blood cells. Their abundance also helps to outcompete against harmful bacteria. For example, Streptococcus pyogenes, which can cause strep throat, is found normally in your throat, but it competes with many other bacterial species, that it doesn't really become a problem to us under normal conditions. Only when an abundance of strep bacteria is introduced does the disease start. Our skin also has lots of prokaryotes, even specific species depending on the body region. The underarms have different species than those found on your head. And while little is known about the benefits of skin prokaryotes, a few studies have shown that some beneficial prokaryotes on the skin secrete chemicals that prevent the growth of certain pathogenic bacteria. What we do know is that your brand, or body odor, is sourced by bacteria that break down organic molecules you secrete from your skin. So hopefully by now you've seen the importance of life unseen and can appreciate the benefits and even damages that they can cause. In our next set of videos, we will move on to domain eukarya. But first, where do eukaryotic cells come from?